Yesterday, I showed you some slides where I talked about different contrasts that you can do when you do your hypothesis testing. But I actually didn't put numeric values or anything. I just sort of I talked about it in general terms. So this morning, I actually went in and assigned values that approximate the expression that I'm showing in this particular experiment, where I have the two sexes, male and female, and then three conditions: low, moderate, high, maybe drug exposure or something. And so if you do hypothesis testing on sex, it turns out that that difference is highly significant. There's no overlap between the female and the male samples. In fact, the way I set it up, the p-value was like 10 to the minus 80 or something. So that's not in question. And then you might also ask, okay, is there a significant difference among the categories of the exposure here? So low, moderate, high, and you can see a priori that you see a lot of variance here, slightly less variance here, whereas here they're similar, and so that may affect your analysis. It turns out, actually, if you do a condition-specific analysis without fitting sex in the models, that actually you get no significant difference among the conditions. Okay, so even though it's pretty clear that the average difference is a little bit higher in females, because as you can see, it's certainly higher in females across the conditions, but it's so similar in the males that actually you get no overall condition effect. But if we model them jointly, so in other words, if you actually go in and you actually fit an analysis of variance model where you've actually got both sex and condition in the same analysis, it turns out that adjusting for sex in this particular version allows you to see that the conditions are actually significantly different. Okay. And then the next step beyond that is to actually ask whether or not there's an interaction effect, which we'll get to in a minute. Another thing that you'll see typically people do in the literature is to just go ahead and say, well, actually, I'm not going to do a comparison of males and females or conditions to conditions. I'm going to compare just all of the 6 possible groups to one another. And if you do that in the analysis of variance framework, then that's extremely highly significant as well, as you might expect in this particular case. And then a heck of a lot of literature you'll see in immunology literature and sort of people who are less used to complex designs. People sort of say, well, okay, they'll draw up histograms of effects and say with asterisks representing moderate, strong, or very strong significance, sort of joining each sample to each other one. So I don't generally recommend that approach, sort of a one-to-one comparison of all possible comparisons, but you'll see that in the literature quite a bit. And then, of course, the post-hoc contrast in this particular case would be highly significant. But what I mentioned at the end of that lecture was you can also evaluate these interaction effects. And so it turns out if we fit a model, a fixed effect model, where we have sex as being a fixed effect and these conditions as being a fixed effect, and then you evaluate it as an interaction, in other words, whether or not that slope is significant, it turns out that that interaction effect is highly significant in this particular case. But if instead I turn around and say, well, actually, what if the condition itself is a random effect, which makes the interaction effect a random effect as well, so I can't actually specify the significance of it, but what that does is actually if we model these as random effects, it turns out even the sex difference is not different in this toy example. So just by way of sort of pointing out to you that the way you specify your model really has a very important impact on what you're going to kind of find at the end of the analysis. And so having in mind exactly what it is you're looking at and what potential covariates are and whether they're fixed or random really should impact your output. But I have to say that in my opinion, a heck of a lot of literature really doesn't pay a lot of attention to that, partly because it really wasn't built into the main tools that JAR and DE seek, although I think they're much more conducive to that now than they used to be. Okay, so I'm just going to stop that lecture there, and now we'll start on the normalization issue. Okay, so what I'm going to talk about now applies much, much more to bulk RNA-seq than to single-cell RNA-seq. In fact, there's not much attention paid to normalization issues in single-cell as yet at all. So really think about this in the context of bulk RNA-seq. And what I want to do is really explain both how we normalize data and why it matters, both statistically and biologically. So the first thing you may have noticed or asked yourself is, why do we work on the log base 2 scale? So whether or not it's microarray data or bulk RNA-seq data, or in fact even single-cell RNA-seq data, what we're usually doing is taking our counts, and they're usually normalized to counts per million, and then we log transform them. Okay. So what a log transformation does on any scale is actually make the data more normally distributed. Okay. And what that does is minimizes biases that are due to the common feature that a small number of genes account for over half of the transcripts in a study. I've got to go to the next slide so you see that here. Okay, so this is just a data set I downloaded from my computer that I had from a few years ago. It happens to be a human blood data set, and this is counts per million. And actually, I've truncated it even at the far end here, just for purpose of illustration. So what you see is that there's a small number of genes, so it looks like a high density, but it's a couple of hundred genes, which have very high counts per million. And so they actually account for about half of the reads in the entire data set. The vast majority of genes are actually down here in this low abundance set, okay? If we take the log transformation, then you can see that it's not a perfectly normal distribution anymore, but it's more normal than it used to be. But what you also see now is that you've actually got equivalence of fold differences. So you often say it's something twofold or threefold or fourfold difference. And so any number difference on a log base 2 scale corresponds to a twofold difference. So 4 versus 5 is twofold. So 16 is half of 32, 32 is half of 16 counts, and log base 2 of 32 is 5, log base 2 of 16 is 4, log base 2 of 64 is 6, okay? So the difference between 4 and 6, or 6 minus 4, is actually fourfold difference, okay? And then that's a eightfold difference out to 7. So it's very, very symmetric measure. So the reason why I use log base 2 is because it's very convenient in practice. It turns out that at least among strains of organisms, most differential expression is in the range of 1.2-fold to 10-fold, something in that range. Now, if I'm comparing retina to liver, of course, you're going to see differences in gene expression that are 100-fold. But most applications, you're really interested in the effect of a treatment on the same tissue, okay? So in that sense, working on the log base 2 scale is by far the most convenient, because it means that numbers that sort of range from 2 to 10 are in that range where you're capturing most of the expression differences, okay? So you just need to think of 1 as 2-fold, 2 is 4-fold, 4 is 8-fold, and so forth. And that's true whether it's negative or positive. So intuitively, it's actually simpler to infer fold changes in a symmetrical manner. The difference of -1 unit corresponds to half the abundance. The difference of +1 is twice the abundance. The difference of -2 is a quarter, of +3 is 8 times, and so forth, okay? So it's just very, very easy to think about it that way. It also happens to be that the log scale is insensitive to mean centering. So you can just center your samples. You make the mean of your sample after log transformation 0. So negative value is less than the average, positive value is more than the average, but that has no impact on this idea that the value of 1 corresponds to 2-fold. So it preserves that relative abundance above or below. And then one other point I need to make is that you'll often see that people add 1 to all values to the raw counts before they take the log. And the reason for that is that it avoids returning a null value. So this symbol here, if you do it in Excel, because the log of 0 is a meaningless number. Okay, so you just add 1, and if the raw values in counts per million are between 1 and 5,000, then adding 1 is not going to make any difference at all. Okay, but that step is typically built into most code. I should add that actually earlier versions of Seurat, which is the main single cell processing software, actually took log on the natural log scale instead of log 2. One of my students noticed that, which I think has been corrected, but I don't know why they did that. All right, so I showed you this slide. So we have log base 2. It's a very convenient scale. It's you still got the majority of transcripts at the low expression level, but things are much more symmetric. Okay, so this is what we call the total frequency distribution of all transcripts in a sample. Okay. The other thing that's worth talking about is what this negative binomial distribution is. And what it applies to is really only the very low abundance transcripts. Because a negative binomial distribution that you see at the top here is a typical negative binomial distribution is what you expect to see when there are failures. Okay, so you're modeling the probability that some outcome is not observed. So in this case, that outcome is any measure of gene expression. Okay, so if you've got a low abundance transcript, there's a chance that it's not going to be expressed in the sample. And so you get a lot of what are called zero counts. Well, if you repeated that experiment, then it's quite likely that you would see some sort of transcript. So if you see it twice and then you add up the values across the two, then actually your distribution will shift to this. And if you do 4, it becomes more normal. So what a negative binomial adjustment is doing is adjusting for the fact that at the low end of the spectrum, the reads are actually not normally distributed. So if you take a gene out here with a average log 2 CPM of 7 or something, then they're actually going to look pretty much normally distributed. And then your T statistics and your F statistics are very valid. But if you're down here at the lower end, then that distribution is not normal. And so you have to adjust for that. And so what you're doing is adjusting for the expectation that the distribution is in fact negatively binomially distributed. One other thing I should ask is why is it counts per million rather than counts per 10,000 or something? And it's just because it's a convenient number. It doesn't matter. It could be counts per 10 million. But it just brings us into a scale that's pretty usually giving us these sorts of numbers that people are familiar working with. The single cell data, we actually typically transform to counts per 50,000 is often seen, or maybe counts per 100,000. And that's because the read depth per cell is much lower, or even counts per 10,000. But what you want is some common number so that all of the cells are on the same scale. Somebody's, I'm getting feedback. Maybe everybody mutes. It'd be good. There's a chat question there. So yeah, I mean, there's this big advantage is that it's easily interpreted and it's not susceptible to the effect of outliers in your distribution. If you're just working on the raw scale, then looking at correlations, things like that are really affected by your outliers. And there's something else I wanted to say that I'm forgetting, but maybe I'll come back to it. Okay. So in the microarray days, we generally did just a simple additive adjustment to center the mean or the median of the distribution. So we'd have our log CPM counts. Well, we didn't have CPM counts in those days. You just have your frequency distribution of counts. And then what you want to be sure of is that the mean value for all of your distributions is very similar. So what we used to do, so if these were two distributions, a blue one and a red one were just two different microarrays, in the old days, you just shifted the blue upwards or the red one downwards until they overlay on top of each other. And if they're exactly the same distribution, then they would overlay perfectly. And so that was just a simple additive shift. When RNA-seq took over, the impact of actually doing your multiplicative scaling, which is this counts per million scaling, is more like sort of grabbing the tail of this blue one and pulling it until the mean is the same. Okay, and so that means that actually the two distributions in this particular case would no longer overlay one another. And I'm not here to say that one is better than the other. It's just that you should be aware that there are different consequences, some slight consequences of doing that. The other important thing, which I did mention yesterday, is there's an adjustment called trim mean of the medians, which is accounting for biases due to the variable abundance of this small number of highly expressed transcripts, like hemoglobin and ribosomals and so forth. So what this procedure does is it takes the upper and the lower, I think it's decile or some fraction of the reads, and it discounts them in the normalization. And effectively, that means that what you're doing is you're doing the normalization on the bulk of the transcripts that are in that range where they are expressed, but they're not too highly expressed. And so the normalization is not biased by those outliers. Okay. And as I think I said yesterday, if those reads counted 50% of the transcripts in one sample, but 30% in another, then it would mean that the CPM would typically be higher in that second sample. And then we discussed zero counts as well. Okay, so if you read the literature and look at it in detail in EDGAR or DSP manuals, they will actually oftentimes show you these MA plots. So an MA plot is basically the fold difference plotted against the magnitude of effect. So actually, instead of volcano plots, typically EDGAR actually, if you visualize your data, it will produce a plot that looks like this at the end, and then it colors the most significant genes. Okay, there's three things you want to know. One is the fold difference, one is the magnitude of expression, and one is the p-value. So a volcano plot optimizes visualizing the p-value and the fold change. It sort of ignores the actual level of expression, whereas these MA plots allow you to look at the dependency of the fold change on the level of expression. And what you see in this top plot of the raw log-based 2 data is that, so the null expectation is that there's no relationship between fold change and abundance. There's no a priori reason why lowly expressed genes should be more likely to be up or down regulated than highly expressed genes. And if you see a curve, so you can see a sort of the red one there is sort of slightly curved upwards there and down, that you want to remove that effect. Okay, so one of the aims of the normalization is to adjust for that variance in the distribution. Okay, so that at the end, so this M is in fact just the logarithm of the ratio or the difference between the two logarithms, M1 minus M2. So that's the fold change. One is twofold, two is fourfold, and in the opposite direction. And then A is the additive effect, just the average expression in the two samples that you're looking at, or the two classes you're looking at. Okay, so this is done on an array by array basis. And then what you see here, so you'll see common and trend and tag-wise dispersion talked about in the manuals. So this is actually the coefficient of variation of each gene plotted against its average measure. So not the fold change, so just the degree of variation in that transcript. And very typically, you're going to see in gene expression data that there is over dispersion at the low end. So there's much more variance at the low end. Now, you don't usually expect that in general. Generally, the variance scales with the mean in everything. So if you go and build a snowman and you decide that you want to make those snowmen 10 centimeters tall, then they will, and you make 10 of them, they might be 10 centimeters plus or minus a centimeter in height. If you decide you want to make them a meter tall, then they would probably be a meter plus or minus 10 centimeters in height when you're done. Okay, so just in life, in everything, variance tends to scale with the mean. In RNA expression data, particularly RNA-seq data, that's not the case, and the reason is because of this negative binomial effect. You get dropouts. So you've got a lot, seeing 0, 1, 2, or 3 counts really produces a large amount of variance compared to seeing 20, 21, 22, 23. It's a small fraction of variance. Okay, so you get this scaling, and that's a large part of the advantages you get from the general linear modeling, the sort of shrinkage estimation you get that I mentioned yesterday is adjusting for this trend in the dispersion, and you use that then to make better estimates of the gene expression variance in the data set. So each point is one gene, but adjusted for this this trend away from a linear common trend. So that's what we're looking at when they're describing manual vignettes, what the software is doing. All right, so I want to sort of take a step backwards and think about what different types of normalization are doing. Okay, so let's suppose we have 6 RNA-seq data sets, and we obtained 3 of them from female samples and 3 of them from male samples. Doesn't matter what species or what tissue, just male and female samples is our contrast of interest. And I'm going to plot here the cumulative frequency distribution of the transcripts in the sample, okay, after log transformation. So we saw this, we still got this sort of right shifted tail, but the vast result, there's a mean and there's a sort of a slight excess of low values. And this one sample here, this red one here, for whatever reason, had lower counts than the others. So we adjust for that in a number of different ways. So that's really what the objective of normalization is. But if we didn't do that, we can ask the question, well, is there a difference between male and female for a particular gene? So I've just chosen one gene and I'm indicating where it is, what its expression level is relative to these frequency distributions. So it turns out in the male samples, they were here, here, and here. And in the female samples, they were here, here, and here. And even though the female is slightly higher, so each of the 3 is sort of slightly higher than its corresponding blue ones, that difference, that small difference, is probably not going to be statistically significant given the variance among the arrays. So if you just did that test on the raw data, your conclusion would probably be that there's no difference in expression between male and female, okay? So the most aggressive sort of variance transform you can do is something called a quantile normalization. And what a quantile normalization does is says that I expect that the distributions, so the frequency distributions, are identical for all of the samples, okay? And really that's only going to apply if you expect a very small number of changes, okay? So if you only expect a handful of genes out of 10 or 20,000 to change, then what that's saying is you expect that the other 9,990 genes have the same expression in each sample. And if that's your expectation, then a quantile normalization sort of assures that's what's going on. So what a quantile normalization is actually doing is saying it's ranking every gene in every sample, and then it's assigning the same value, so the average value of expression to each rank, okay? And so what you end up with is that the genes will have different ranks in different samples, but for every rank, it's got the same value. So the actual frequency distribution is identical across all of them. And if you do that in this case, so here's now where the red ones have shifted to, here's where the blue ones have shifted to. And now, in fact, if anything, there's a very slightly smaller average red value. But again, it would not be significant by any sort of test. So they're all, given the variance among the red and among the blue, any difference between red and the blue is not significant. And you would again conclude in this case that there was no effect. Just as a side note, quantile normalization was actually initially developed for Affymetrix arrays to actually quantile the probes within a gene. And then it was just applied to whole data sets and became quite popular for a small number of years in the literature. We go back to sort of mid-2000s, and you can see quantile normalization quite a lot. But people started realizing that actually there is really no basis for doing that. It's way too aggressive, that sort of variance transform. And so you don't see it done very much anymore. You certainly don't see it, I don't think, with RNA-seq, with single cell data. And John Story, who I've talked about quite a bit, has analyzed a bunch of experiments and shown that actually quantile normalization can give you really either losing signal or actually inducing false positives at a very high rate if you do simulated data sets. So, and the reason for that is actually because it's rare in biology that only a small number of the factors are significantly differentially expressed. Okay? Particularly in bulk RNA data sets, we know you've got a large mixture of cell types. or you've got conditions that are actually having an impact, not just on 2 or 3 genes, but having an impact on thousands of genes. Because of that, we actually expect the distributions to be quite different among samples, okay? So if you go to the other extreme of theorizing about how to normalize data, then a very supervised approach will be to say, you know what, I expect that the male and the female profiles are quite different from each other, but that most of the female samples are similar and most of the male samples are similar. So I'm actually going to do my normalization with that in mind. And I'm not going to do this very, very aggressive variance transform. I'm going to accept that there's differences in variance among the data sets. And if I do that, it's much more like just simply scaling to the mean centering and then doing a sort of a variance transform just within the sample types. So supervised normalization is really designed to maximize your power to see differences among samples on the supposition that those samples actually resemble one another more than you'd expect if they were drawn from the same distribution, okay? So now in that case, what would happen if you do the analysis is that now all of my red samples are out here and my blue samples are down here. And very, very clearly, that would be a highly significant result. So you could conclude from this supervised normalization that you actually have a highly significant effect, okay? So so nobody wants to do the raw data analysis, but 2 extremes of analysis are to do quantile and supervised, and they're going to give you quite different results. So in this case, no effect, and in this case, a large effect. And you should be asking yourself, which one is the right way to do it? And actually, I would argue that there is no necessarily correct way to do it. I'm inclined towards supervised, but a lot of analysts have a problem with that because they actually say, well, then doesn't your analysis depend on what factors you decide to supervise and normalize to? And I say, well, that's true, but actually not adjusting for those factors is ignoring that they're there and is not an appropriate strategy either, okay? So there are debates in the literature as to which ones are most appropriate, and then there are possibilities of doing it sort of in between that. The other thing I would emphasize in this slide is that actually any sort of variance transformation is tending to make your analysis on the relative scale, okay? So you're really asking whether there's been a relative shift in gene expression of a gene compared to all other genes. Whereas these more supervised approaches are much more likely to be giving you analysis on the absolute scale, okay? This will be more like a rank order comparison, whereas this is more like actually saying, well, I actually do care whether it's a 2-fold or a 3-fold or a half-fold difference. Those actual raw differences make a big difference on this scale, okay? And as to which genes relative versus absolute differences are important, that's probably going to depend on the type of gene. So at the end of the day, I just don't think there is one absolutely correct way of normalizing data. So I always encourage my students to normalize things in 2 or 3 different ways and look for consistencies or inconsistencies and help them interpret the data. Okay. And so I'll talk a little bit more about supervised normalization in the next few slides, how it's done. All right, so then are the different approaches to normalization, so that you can have median or mean transformation. So that's simply centering the distribution and additive shift. And doing something like that is the minimum you absolutely want to do to control for overall distributional effects. It's like just fitting an ANOVA to the total distribution before you do the gene-specific analyses. Variance transforms, as I mentioned, come in various types. So the simple sort of variance transform would just be to standardize your data. So in other words, not only do you center them, but you actually make the distribution around that center itself normally distributed. And people sort of erroneously sort of thought that that was a good idea to do that, because you've probably heard in statistics that the assumption is that your data is normally distributed. What you have to bear in mind is that your hypothesis testing isn't on the total distribution, it's on the individual genes. So what matters is the actual residuals of the gene expressions, or the actual gene expression measures themselves, are normally distributed within a gene across samples. It's not that the distribution of a single sample is normal. It's that the genes across sample are normal individual genes, okay? there's not a strong rationale for doing standardization. So that's, in other words, converting the distribution as close as you can to a normal distribution. An interquartile range to me makes more sense. So what that's doing is saying, okay, I'll do my mean centering, and then I'm going to set the 25th percentile and the 75th percentile to be the same. And so you sort of squish your distributions into that range, and then you have variation around that. Okay, so that's sort of a mild variance transform, so not as aggressive as standardization. A quantile normalization is the one that I just showed you on the previous slide, is the most aggressive variance transform you can do. And so what it's doing is transforming the ranks to the average expression of that rank. And the result is that the profile, the sort of the total distribution for every sample is identical. Okay. You can avoid doing any sort of transform at all on the total data set. So if you're going to do that, you can actually do gene level model fitting. And if you're doing that, you fit technical and biological effects of interest into your statistical model, gene by gene by gene. Okay. And ideally, that will actually come up with the sort of same result as doing a transformation beforehand. But it's not very efficient. It's not statistically efficient, and it fails to account for the very strong covariance of expression of genes. Okay, so you can avoid this. So what all of this is doing is basically trying to adjust for the fact that there are technical factors like batch effects and RNA quality effects, and that there are biological factors such as sex differences that lead to the differences being different. So you can actually fit those factors into the gene level models without normalizing them here. But as I just said, it's not very efficient, and it's probably not, well, certainly not as efficient at controlling those effects. And then the final one that I mentioned there is supervised normalization. I'll go into that a little bit more detail in coming slides, but it's really optimally estimating whichever the biological effects you care about are. And you try to not remove those, but at the same time to remove the technical factors across the entire experiment. Okay, so there are methods like variable analysis, which Jeff Lee developed. So he developed combat, which is the most common method for removing batch effects. But an iteration of that is something called surrogate variable analysis, which is basically where you identify the technical factors of interest and you remove them from the data set just by linear regression. And then you go ahead and do your analysis on what's left over. I'll also talk a little bit about something called SNM, or supervised normalization of microarrays, which is a very iterative cycling of that. I'm actually, since it's not used as much in RNA-seq, I'm not going to go through it in any detail. We used to go through it. John used to go through it because he developed it in a lot of detail, but unfortunately, it's not used much anymore, and he never developed it specifically for RNA-seq data. So I actually won't go into it in a lot of detail, but I'll mention it because I actually like my students to do it on occasion. SVA has been written for RNA-seq data. So you can look, if you go to Bioconductor, look up for a package called SVA-seq for doing surrogate variable analysis. All right, so what impact does this have? So this is, just suppose these are raw profiles. This is from microarray data, but there's no reason why you shouldn't see differences like that from RNA-seq data. So, in the old days, we could do something like a median transform, which would just be, it's not a variance transform, okay, it's a median transform, or a mean centering transform, would cause the data to start looking like this. And in this case, actually, that transform is almost like a natural IQR transformation, because this 20th and 75th percentiles, so they wouldn't exactly be the 25th and 75th. But you can see that there's a crossing of those values approximately at the quarter and the 3/4 mark. Okay, but you haven't done anything to flatten, this one, which has fairly low dispersion. One interesting thing you can do is, if you do this analysis, you can actually go in and ask the question whether or not there's a mean difference in dispersion of the red and the blue samples, which is actually an upfront thing you can do, is actually to confirm that actually males and females are different. So if the blue ones and red ones are males and females, in this particular case, there may not be a significant difference, although perhaps there's a trend that the blue ones are less dispersed than the red ones. And so knowing that actually can help you decide what normalization approach to take. But another way of representing this data then is to plot the means of the sample or the median. Actually, I think this is the median. Well, no, I've transformed to the median, which is 0. So these are the means. Then they have a variance around them. And you can see that that's not being taken care of. So you've got some samples which have fairly low variance. So in other words, there's lower dispersion between the lowest and the highest abundance transcript, whereas other ones are much more spread out. And a priori, it's not necessarily really clear as to whether or not that's a biological effect or a technical one, but there are good reasons to expect that biological factors cause differences in dispersion. Okay. And then I also said to you that CPM transformation is not so much doing an additive shift, it's just sort of pulling them, so doing a multiplicative shift, but it has the same impact of centering the data. All right, and then if you want to go ahead and do variance scaling, what that's now doing is sort of taking that variation in the means or the medians, and it actually is creating very, very consistent variance around that, whether you do it by standardization or by IQR or by some other method. So you're left with these outlier genes, which are very high abundance transcripts, explaining a lot of the factors. And then you've got the vast majority of transcripts down here. And then you've got methods. So one of the things that EdgeR and DSeq do is they actually do a factor. They create a scaling factor. And in EdgeR, it uses this TMM method to actually adjust for the proportion of the reads due to these outliers. Okay, so their transform is based on identifying a factor that's used to sort of optimally adjust between these variance and mean centering approaches. Okay, so if you really step back another step and ask, well, what's the challenge in normalization? I mean, this is a figure I stole from John Story a few years back that really, I think, illustrates it quite beautifully. Okay, so instead of doing a red-blue coloring, he's just done it yellow to green scaling. So yellow would be high abundance and green low abundance. And what you've got is actually a 2x2 design in this case. So he's actually got, say, males on the left and females on the right. And then within those, he's got 2 treatment groups. So he's got control and disease. So first 3 would be control, second 3 would be disease, first 3 would be control, second 3 would be disease. And so we've got healthy male, healthy female, diseased female. And the idea is that what you're seeing there is made up of 3 different types of variation, okay? So one is the actual biological signal. So if I could actually do the experiment absolutely perfectly, measure everything perfectly without error, then the true result, which is what you're trying to get to, is shown here, that in fact, within the male samples, there's absolutely no effect of disease whatsoever. But within the female samples, you get a big effect of disease. And in fact, you get a very, very large difference between the males and females as well. In fact, in this particular case, the male samples tend to be low expression. The genes that are low expressed in male tend to be high in females and vice versa. But you do have this big disease effect where you've got a whole bunch of lowly expressed genes in healthy controls that get turned on to some intermediate level in the disease state, okay? So what's getting in the way of us being able to visualize these biological sources of variation is the fact that, in fact, there is just stochastic noise, just due to labeling, due to just specificity of the sample you had to get, due to a whole bunch of different reasons. And then less so much with RNA-seq data, but it's still there, but this is particularly strong in array data in the old days. You've also got technical sources of noise, okay? So it turns out, if you look at this, you can identify some samples. So maybe the first 2 here in the diseased female, maybe the first one here in the healthy female, and maybe this one here, first one of the diseased male, all tend to have higher abundance of these genes, of a lot of the genes, at least the ones we've chosen here, for some reason. Okay, so that might be just related to RNA integrity. So maybe there was just less degradation in those samples than the other ones, and so you're tending to get higher abundance measures, okay? So that would be a hidden covariate, or maybe there was a batch effect of some sort, that those samples were all processed together. And if you know that, then that's great. but oftentimes we just don't know what those hidden sources of noise are. Okay, so that's our problem. Is we're trying to identify this, but we're doing it in the presence of technical noise and stochastic variance. And so the whole approach of a supervised normalization strategy is to say, can I identify the genes that are affected by the technical noise and adjust for that to the entire array without affecting the genes that that I know there's a strong biological effect. Okay? So the SNM procedures and the SVA procedures, I won't go through the algorithms, are doing just that. Okay, so you're trying to preserve your biological noise while removing the technical noise, which gives you a better chance of seeing this. And then your stochastic variation is just taken care of in these approaches, sort of that adjust the array effect overall. So you're not overall biased by factors specific to that individual array. Okay, but you're always going to have residual variance unexplained. The idea is we can identify technical factors from the data. We can fit them, if you like, in a regression framework, and we're left, hopefully, with a better representation of the true biological signal. So that's the challenge. And if you don't recognize that and you just analyze the data in this, then you're going to be less well-powered to find things than if you do this procedure. That's really, in a nutshell, why normalization matters, and why it is that if you apply a supervised normalization strategy, you can oftentimes see things you wouldn't otherwise. Remember, a few years back, I was teaching this course. I had a student who had several hundred samples she'd collected, looking at arsenic exposure in, I think, blood samples in a Bangladesh population she was working on. And she'd done her analysis and seen absolutely nothing significant. And we actually sat down that night, and I was able to process her data through this framework and actually identify several hundred genes that were differentially expressed when you took into account this, and they actually fell into pathways that made sense with respect to the biology. So it really is worth sort of looking at these procedures to see if you can find things that are hidden by the fact that in some data sets there's a lot of technical error. I've talked about surrogate variables. They're not quite the same as, but they're sort of similar motivation as principal components. So I'm not sure how many of you in this module know what a principal component is. So I'm just going to try to give a very lay explanation of what principal components are. So imagine you have this class, and we were all to sort of just do a survey and submit a bunch of attributes. So we might submit our height, and we might submit our hair color, we might submit our hair length, we might submit our skin color, we might submit our eye color, and just a whole bunch of things like that. Well, if we process that data, we would find that those measures were not really independent, okay? So we'd see a difference in waist to hip ratio that was correlated with height, and we might find that height is correlated with hair length. And after the fact, we could say, well, that's because male and female samples tend to be different. Now, of course, not all males have short hair and not all females have long hair, but there's a trend there, and that causes covariance. Similarly, we might find that hair color and eye color and skin color are correlated to some extent. So we know after the fact that Asian or African ancestry individuals tend to have darker skin and darker hair color than, say, Northern European. So what principal component analysis is doing would be taking data sets and in a very systematic manner, finding the sets, so components that explain a large amount of the variance. So PC1 is that set of correlation structure in the data that explains the largest amount of the variance. So you fit that, and then having fit that, we can go and see what's the second biggest component that's residual. So it may be the first principal component of those things I just mentioned was in fact sex or gender. And then the second one might be ancestry, and then the third one might be smoking status. Who knows? Depends on the data set. Okay. As I mentioned right at the start of this lecture, I said that these things can be very much data set specific. Okay. And it's true of principal component analysis as well, that the outcomes tend to be very much data set specific because they're a function of what types of and ratios of individuals you have in the study. Okay. But a really good thing to do with any gene expression data set that you get is to start out by doing what was called principal component variance analysis. And what it's doing is allowing you to see what factors in the analysis are explaining most of the variance. So another method that's very similar and related to this is something called multidimensional scaling. This is in fact a multidimensional scaling plot, but it's pretty much the same as a principal component analysis. And I had an experiment that we used to analyze in the course, but I'll just show it here, which was consisting of, I think it's 4 African-American women in normal weight range or lean weight range, 4 European-American women in lean weight range, 4 African-American obese, and 4 European-American obese. And so these are just individuals from a study that we were doing in part of just a very small part of a very large study we've been doing on the effect of health factors on gene expression in peripheral blood. And so if you just analyze those 4 women of each of the 4 categories, and you do the principal component analysis on gene expression. Okay, so now it's PCs on 10,000 genes rather than just a half dozen factors, but it's on 10,000 genes. PC1 or dimension 1 of multidimensional scaling captures some portion of the variance. And in gene expression, that's typically going to be maybe 20 or 30% of the variance. And then PC2 is maybe going to be 5 or 10% of the variance. Okay. And so then you can plot the factors, and then you can just visually ask what do they correspond to. And so if you look at this, you might see, okay, well, let's look at O and L. So actually on the left-hand side here, with the exception of that one and a couple of these, the L, which is lean, tend to be the left, and all of these ones to the right, the O, and even down here, the O tends to be the right. So it looks like PC1 is actually capturing the effect of obese versus lean. And then you might look further and say, well, where are the African European? And it turns out the E down, there's exceptions obviously, but the E's tend to be at the bottom, and the F and the A's tend to be higher at the top. So actually PC2 tends to capture the effect of ancestry, which we used to code as ethnicity over here. But it's ancestry effect. So now we know then that PC1 is capturing the biological effect we care about, which is metabolic status, obese and lean. And that's also in an experiment where actually ancestry matters as well. And PC2 captures that. So we've already learned something about the data without actually having to do tests at the individual gene level. Okay, it could have been that actually PC1 was date stamped on the arrays, which is a batch effect. It could have been that PC2 was the effect of RNA quality. In this case, it wasn't. It's actually 2 biological effects, but it could be that. And it's good to know what that is before you normalize the data. So there's software out there, actually PVCA software actually happens to be been written by a former student of mine, Pierre Buchel, who was at the NIEHS for many years. It's now, like so many people, moved to the industry last year. But he wrote this vignette that you can find on Bioconductor, but you can find it in other software groups. I think actually Seurat might also have a version of it. But what it's doing is saying, okay, I'm going to identify all of my principal components, and I'm going to ask how much the variation in that PC is explained by various factors of interest. Okay, so those factors in my case are ancestry, obesity, RNA quality, batch, and so forth. And so what we get is then a weighted estimate of the proportion of variance explained by those. So maybe 50% of PC1 is obesity, but only 5% of PC2 is obesity and 1% of PC3. So I add up those proportional contributions of those PCs, and I get an overall estimate of the combined effect of obesity. I can do the same thing for ancestry. So in that case, it's only maybe 5% of PC1. So it's 5% times whatever, 40% of the variance is explained by PC1, but it's 50% of PC2, which explains 20%. So 50-20 is another 10%. So it goes through that weighting strategy. And in this particular case, it turns out, even though PC1 is metabolic status and PC2 is ancestry, when we weight them across the first 5 principal components, it turns out that ancestry and body weight actually both explain around about 15% of the data, 10 to 15%, so it's 11 and 14. And then actually I include an interaction term in this analysis. I'm actually able to evaluate that actually the combination of weight by obesity is actually explaining another 13% of the data. So you add those all up, you get, what's that, 25, 35, 38, or 39, and then the residual there, or 63%, is unexplained variance, okay? So PVCA analysis is something really, I think, good to do straight up, because it gives you an overall picture of what's causing the covariance of gene expression in the study. And if you actually, so here's an example where we've done it. In this particular study, we actually have ancestry, weight, the visit. So I didn't, so an extended data set, I've actually got each person measured at 3 different times, and I've got person in this. And when I do that, so it's not the same as the previous data set because I've actually added visit to this one. So what this is telling me that actually is that PC2, or the second fit, is actually highly correlated with person. So a lot of the variation that we saw in PC2 in this extended analysis, because I've got visits as a degree of freedom to evaluate that, is actually differences among the individuals. Whereas when I go to PC3 now, in that extended data analysis, it turns out that ethnicity and individual are both correlated with that, okay? So surrogate variable analysis is like PCA, but it's only doing it on those subsets of genes which are variable. And it's doing it from the data. You can just use the software. You can read the vignette to sort of work out more about it. I don't want to go into details, but it's a really useful tool. And if you run that, so if you do your surrogate variable analysis, you find out what those surrogate variables are, and then you ask, what are they associated with? And if you find that the first one or the second one or the third one is associated with something technical, like batch or like RNA quality, then you probably want to go ahead and remove that effect from your data set just by regression, leaving behind residuals, which are much more likely to be affected by the biological factors. So that's the very simplest thing you can do. So SVA allows you to do that. There's another similar tool called peer factor analysis. PER is a Bayesian method. I tend to be frequentist about where I approach things, so I use SVA, but peer factor is a Bayesian form. Works similarly, although by default, peer factor actually fits all of the factors and removes them. And that was because peer factor analysis was developed actually to support EQTL analysis, where you want to get rid of as many of the covariates as you possibly can, so you're left with just studies of the effect of genetics on the gene expression, okay? So peer factor analysis tends to be very, very aggressive about removing all covariates, whereas SVA and then SNM, which is this iterative version of SVA, allows you just to remove ones that you know are representing technical variates, so you're left with the biology. You may also find that maybe surrogate variable 3 does not associate with biology or technical, and then make the decision to remove that anyway because it's not, maybe there's a technical factor there that you haven't accounted for. You don't know what it is, but it's nevertheless there. And then combat is like a reduced version of SVA that just is there deliberately for batch correction. So you're not actually identifying the peer factors. You're just directly removing the batch effect, okay? So really good approaches. I should say for single cell data, not recommended to use combat. It doesn't work well. You get weird distributions like that. But for bulk RNA-seq data, combat's very, very widely used just to remove known technical and batch effects. SVA is used to additionally remove hidden confounders or to remove biological variables that you don't want in the analysis. And then this method, SNM, is used, is sort of superior, at least for microarray data, but we use it on RNA-seq as well. Okay, and then does it matter? So this is just a slide sort of showing that really it does matter. So this is Q values, or false discovery rates values, but they're, just think of them as P values for your purposes. So if we just plot P values of raw versus combat, you can see there's quite a lot of dispersion, although they're reasonably similar down here at this end. But if we honed in, we'd actually see hundreds of differences down there. It's actually clearer to see if we compare, say, surrogate variable analysis on a data set against raw. And so what you want to be doing is down here. So anything below the diagonal is genes that are doing better in SVA, getting better estimates than raw. So there's a huge trend for the SVA to sort of give you higher significance, but also you'll see that there's quite a few genes where SVA is showing you that there was inflation of the test statistics of false positives in the raw data. And even SVA versus combat is giving quite large differences. So you want to be down here, but what I'm saying is that you can see just by comparing test statistics that normalization up front can have a really big impact on your data analysis. If you just want to visualize what it's doing to the distributions of the data, just to help you sort of see that. So the quantile normalization, this is another small human data set that Peter Chin, a master's student, looked at some years ago just to highlight this. So quantile normalization, as I said, transforms the total distributions to be the same. So if you have the raw data, if you do a mean centering, it looks like that. If you do an in this particular data set, IQR, it looks pretty good. I mean, if I had data distributed like that, I'd think that's, it's not as aggressive as this, but that looks pretty good. Another thing we did is said, let's just normalize by fitting the date of the experiment and the RNA quality, the RNA integrity. So that produced something that's still pretty dispersed. I really wouldn't want to do that. In this particular case, we fit our ratios of lymphocytes to monocytes to neutrophils. So we knew that, so you can go online, like the Framingham heart study data set, they've already done that. So they just removed the effects of lymphocytes, monocytes, neutrophils from their bulk RNA sequencing data. So if you're analyzing it, you have to be aware that they've actually done that. SNM is sort of somewhere between IQR and these other transforms here. So it's certainly got less dispersion than mean centering. And then here I've just fit all of the principal components, the first 16 principal components to try to remove everything, sort of more like Peer Factor would doing. I haven't gone ahead and just also mean centered those, but just fitting the PCs leaves you something like that. Then what impact does that have on the correlation structure? So these are just heat maps where what I'm doing is the red square means that the overall distribution is much more similar. So it's the correlation of the principal components of the array. So red is similar, blue is dissimilar. So comparing raw to mean centered, there's no impact of that because all you've done is sort of a mean centering shift. Quantile normalization, you've still got some residual similarity, but there's no gross structure to that, which looks good. Principal component analysis, we've actually removed completely the correlation structure. So that's what you'd expect to happen. And then these other methods get something intermediate between those. So even in this case, what the SNM has done is actually produced a pretty high correlation structure among subsets of the arrays, which it was designed to do, to actually find the things that are biologically most similar. Okay, so what do I recommend doing in terms of analyzing data sets? Well, the first thing I'm saying is normalizing the samples matters. And I always encourage people to pay attention to the overall distribution of the profiles to get a sense of whether or not they've done it correctly. And you can either do that by box and whisker plots, or you can do it by just looking at the overall frequency distributions, okay? So it's really a good sign that you've normalized the data reasonably. But just doing that, so the baseline methods in EDGAR and DEC are not supervisory methods, okay? So there's applying an algorithm to all of the arrays. They're not doing anything like SNM does or SVA does, which is to try to do a supervised normalization, okay? So to do supervised stuff, what I recommend doing is extracting the principal components of gene expression or the surrogate variables and asking whether the major principal components are correlated with those technical variates like batch or RNA quality. Sometimes they're not going to correlate with anything. You can decide whether or not to remove them. But what you don't want to do is sort of to throw the baby out with the bathwater. So the problem with sort of just removing all of the correlation structure with all the PC is that you will actually remove biological variables of interest. Okay, so you don't want to remove the effect of sex or to remove the effect of ancestry, by fitting all the principal components, because then you're just going to throw away the signal you're looking for. Okay, so the whole art of this is to remove the technical effects, leaving the biological ones there. Okay, and peer factor analysis in its raw form by removing all PCs or all correlated structures, actually also removes a lot of the biological signal. So you've got to be careful not to do that. Okay, so but if there are PCs there that are associated with batch effects and technical effects, then you want to remove those by some sort of regression strategy. And then the other thing, point 4 here, that I said is that I sort of do encourage my students as much as possible to analyze data sets in several different ways with different methods. And so what that does is two things. It confirms that the findings are not sensitive to your analytical choice. Okay. So, and if the findings are different as a function of analytical choice, then oftentimes that will help you gain insight into what's causing those differences. So one thing you might find is that those differences reflect difference between absolute and relative scale transformations, which is telling you perhaps about whether or not the different lists of genes you're looking at are important because they're relative or absolute differences. Or maybe doing these comparisons of two different methods actually helps you to find what confounding factor was. So maybe unsupervised versus supervised actually lets you see that a big factor in analysis was confounding date of hybridization or date of sampling or something. Okay. And then once you've done all of these methods, once you're satisfied that you've normalized your data set, you've analyzed it with the different DE methods, you compare your final p-value distributions and then do downstream analysis, things like gene ontology analysis on those to identify the biological pathways that are in common, that are differentially expressed. And we're not going to cover gene ontology in this module. If you're interested in that, then I recommend module 16. I think I have one more slide in this section, a little bit more about biological interpretation. So as I said, variance transforms tend to focus the attention on relative changes of abundance, but it's not really clear whether biologically, whether it matters to a cell, whether it's the ratio of gene A to gene B that matters so much to the absolute levels of expression. Okay, so does it matter that in healthy samples, gene A has increased relative to most of the other genes in the genome, or does it matter that gene A is actually 10 transcripts per cell versus 5 transcripts per cell? They're different questions, and different methods give you different answers. And it probably matters differently for enzymes and kinases, which I would expect would be, or enzymes may be important for absolute levels, kinases relative levels might be more important. Cytoskeletal factors may be absolute levels, growth factors may be the other. Who knows? But different methods are going to give you different results. Another thing I try to emphasize is that there's no reason to assume that all samples have the same overall distribution of transcript abundance. If you do something really aggressive like quantile, then that's your assumption, but it just doesn't fit biology. Okay. I think almost all the other approaches that are commonly used, except that there's pervasive covariance. It's just that supervised approaches are assuming that biological similar samples are transcriptionally more similar, and it does the adjustment with that in mind. But what some people are uncomfortable with is that in fact implies that different analysis pipelines are going to yield different results. And that's certainly true. If I keep sex constant in one analysis and keep drug exposure constant in another analysis, then I will probably get different outcomes. So maybe the right thing to do is to actually keep all of your biological variables of interest constant and then just remove the technical things. Something else that we have to keep in mind is that different genes have different variants. So a lot of the advantages of the general linear modeling framework in EdgeR and DESeq is actually adjusting for that fact that different genes have different tagwise and trends of dispersion, which themselves are related to these technical, biological, and overall abundance effects. So they're the three things we care about. And how you model those then is absolutely going to affect your significance values and even your differential expression estimates, on top of which, as I pointed out, different differential expression methods are going to give you different significant values as well. So it's worth just analyzing your data in different ways to be sure that the things you're focusing on in your paper are as far as possible invariant to the way you've analyzed it. Okay. So inevitably, the choice of analytical strategy will affect the outcome. So in some sense, doing this science is an art more than a science, but I think that's okay. I think good science does require feedback between the biologist and the statistician. How do you know if you've done it right? Well, if you do an analysis, you actually find no genes in common. They're not sharing pathways, they're not sharing functions. That probably means you've done something wrong, because just about every biological factor experiment that I've ever looked at has found genes that are in common. But just relying on the fact that your output makes biological sense is itself a little bit dangerous, because you could suffer from the positivist bias that, oh, I saw what I expected, so what I did must have been correct. And actually, it was interesting listening to Pom this morning in his lecture. He sort of said he actually likes to use, the fact that the hierarchical structures make sense is a sign that he feels like he's done it correctly. And so I'm saying the same thing here, but just with the added proviso that there is a potential for circularity and a positivist bias in doing that. I think that's the end of this lecture on that. Yeah, so absolutely. So Julian has asked, would I recommend doing DSeq2 and EdgeR? I think there's absolutely no harm in doing that. But I would say that within those, I would actually specify different models. So just make sure that you're evaluating different models. Neither of those, as I mentioned, do the supervised approaches. So if you're not getting as much out of your data set as you think you might, or if you do your hierarchical clustering, or you do PCA analysis and you're seeing that there's a really large biological factor in the analysis that you expect to be associated with gene expression, then I recommend doing a procedure like SVA or SNM on top of the DEC and EdgeR analysis. And then the final thing is that none of these approaches are implemented in standard single cell analysis software. So that is totally unsupervised clustering. So I think we're probably not extracting all we can out of single cell data sets so far at all. Okay. But very, very final thought is, if you're an experimentalist, find a statistician friend. And if you're a statistician, be aware that gene expression data sets don't fit the model for other large high throughput data sets. So it's a collaboration that we need. Okay. So it's now 1:40. I'm happy to take any more questions. I'll just stay on another 5 or 10 minutes. But then I'd suggest that we